Now we're going to address the metabolism of nucleotides. Some of the questions you should be able to answer when you're done with this topic are, can cells synthesize nucleotides? How do they synthesize purines? How are purines degraded? How do cells synthesize pyrimidines? And how do cells form the deoxyribonucleotides that are necessary for DNA synthesis? And how are thymine nucleotides synthesized? Sorry, I'm tripping over my tongue this morning. So let's begin with looking at, once again, the central dogma of molecular biology. This basically concerns the flow of information from DNA, which of course is the genetic information storage molecule, to RNA, and then from RNA to protein. The focus of this flow of information is not on the chemical conversion of one form of genetic info to another, but in this sequence, DNA is not converted into RNA, and RNA is not converted into protein. Rather, DNA provides a template for RNA synthesis, and RNA provides a template for protein synthesis. So let's revisit the 5 nitrogenous bases. These constitute almost all of the base material found in DNA and RNA. The 2 purines, adenine and guanine, are found in both DNA and RNA. The pyrimidine, cytosine, is also found in DNA and RNA. Uracil is a component of RNA, but not DNA. And as humans, we have all of the enzymes necessary to synthesize these 5 bases. So as a refresher as well, these are the sugar components of nucleic acids. Remember that RNA contains beta-D ribofuranose, and DNA contains 2-deoxy-beta-D ribofuranose. The difference between the two being the lack of that hydroxyl group on carbon 2 in the deoxy form. We will see that nucleotides containing beta-D ribofuranose are synthesized in a different manner than those containing 2-deoxy-beta-D ribofuranose. All of the precursors to DNA can be synthesized de novo from simple organic and inorganic molecules such as carbon dioxide, the ammonium ion, glutamine, aspartate, and glycine. There's a definite order to the synthesis of nucleotides. The ribonucleotides are made first, with distinct pathways being utilized for the biosynthesis of purines and pyrimidines, and we will look at these separately. The ribonucleotides can then be converted to deoxyribonucleotides by an enzyme called ribonucleotide reductase. This conversion uses NADPH as a reductant. There is one other conversion that occurs at the ribonucleotide level, and that is the conversion of DUMP being methylated to make DTMP. So we're going to look at pyrimidine biosynthesis in 2 phases, or 2 parts. Part 1 being what we're looking at right now. The 6-membered heterocyclic ring is synthesized in a stepwise manner from simple precursors. So in the first step, the amide nitrogen of glutamine and bicarbonate, along with 2 molecules of ATP, are utilized to synthesize carbamyl phosphate. Carbamyl phosphate synthetase 2 is the enzyme that catalyzes this reaction, and if you think back, it is reminiscent of the first step in the urea cycle. Carbamyl phosphate is unique in that it contains both an amide bond and a carboxy phosphate bond, so two high energy bonds. In the second step, so here we have our carbamyl phosphate. Second step, the carbamyl group is transferred to the alpha amino group of aspartate. So we have aspartate coming in, and that amino group then is going to be transferred to aspartate. The enzyme responsible for this reaction is aspartate transcarbamylase. Okay, so we end up with carbamyl aspartate here. This enzyme, I should note here, is the target of feedback inhibition in this pathway and is subject to both positive and negative regulation. In the third step, we're going to convert carbamyl aspartate to dihydroorotate. Dihydroorotate is the enzyme responsible for this reaction, and what occurs is a cyclization of that molecule, which creates a covalent bond between the amide nitrogen of or this group, amide nitrogen, with the carbamyl group and the alpha carboxyl group. So here's the alpha carboxy group, and here's that amide nitrogen of the carbamyl group. In the following step, the cyclic product is going to be oxidized to orotate by an enzyme, dihydroorotate dehydrogenase, that is coenzyme Q dependent. The reaction introduces a double bond into the ring. So the second part of the phase 2 pyrimidine biosynthesis focuses on converting an organic base basically into a nucleotide. And the first thing that has to occur to make this thermodynamically favorable is the ribose ring is phosphorylated. So we're starting with ribose 5-phosphate, and we need ATP and ribose 5-phosphate pyrophosphokinase. So this enzyme is going to transfer phosphate groups to ribose 5-phosphate. The product here, and we see we have addition to carbon number 1, 2 phosphate groups. We get 5' phosphorylribosyl alpha pyrophosphate as a product here. Let's see. So in the second step, the pyrophosphate group from carbon 1 is replaced with an N-glycosidic linkage between orotate and the ribose. So we have the orotate coming in, and it is then linked to carbon 1, and we get those pyrophosphates released. The subsequent cleavage of that anhydride bond, which releases the pyrophosphate, energizes the overall reaction, and the resultant product then is aridodine 5' monophosphate. The final reaction is the decarboxylation of aridodine 5' monophosphate to give us uridine 5' monophosphate. So our first nucleotide, essentially. So this does not show you all of the steps involved in the biosynthesis of the purine ring. Well, the pyrimidine biosynthesis is relatively straightforward, not so for purine biosynthesis. And this is far more complicated with respect to the pathway, and you will not be responsible for the details here. I'm essentially going to summarize what you should know. So number one, the pathway takes place at the nucleotide monophosphate level, and we're starting with ribose 5-phosphate. The reaction pathway starts with ribose 5-phosphate and adds elements in a stepwise manner at the number 1 carbon of the ribose ring until we get to the end product, which is inosine 5' monophosphate, or 5' IMP. So these are the order, or these are the molecules that are added to 5' monophosphate. And this occurs in 11 enzyme catalyzed reactions. And we see that we have glutamine, we have glycine, we have N-formal tetrahydrofolic acid, carbon dioxide, and aspartate. So these molecules are all needed for the conversion of ribose 5-phosphate to 5' NOP. This also requires 4 ATP to energize this pathway. So we have 5' IMP, or inosine monophosphate, and this is then going to be converted to 5' AMP and 5' GMP in 2 separate pathways, each of which require 2 steps. So let's look at the conversion of 5' IMP to 5' AMP first. Again, this is a 2-step pathway, and there is a fusion product here that is not shown, but the first step involves aspartic acid and GTP hydrolysis. So the GTP hydrolysis is going to provide the energy to drive this reaction. Also, we need the phosphorylated ribose. So from the fusion product then, fumarate, which we know to be a citric acid cycle metabolite, is released, leaving behind the amino group of aspartate, and that gives us then our adenosine 5' monophosphate, or 5' AMP. Now, if we look at the other 2-step pathway that generates 5' GMP, 5' IMP is oxidized in an NADH-dependent reaction, which introduces a carbonyl group at the 2 position of the 6-membered ring. The amino group from the amide nitrogen of glutamine will replace that, and again, that product is not shown here, will replace that carbonyl oxygen atom to give 5' GMP. This reaction needs a high energy molecule. In this case, we are utilizing, or the reaction utilizes, the hydrolysis of ATP to AMP and 2 inorganic phosphates. So Now we have our monophosphates, at least 3 of them, and these are then just phosphorylated by nucleoside monophosphate kinase, and this kinase does not distinguish between these. It works on all of them, and of course we're going to need ATP to donate that phosphate group. So we get our diphosphates in all 3 cases, and then there's nucleoside diphosphate kinases that add that 3rd phosphate to generate ATP, GTP, and UTP. We still have to address cytosine triphosphate. Where was I? What was I thinking here? In order to generate CTP, we're going to convert UTP to CTP. We'll need ATP and glutamine. We release glutamate, ADP, and an inorganic phosphate. And this essentially provides the energy for that reaction. Sorry. The amide nitrogen for glutamine is the source of nitrogen that we need to generate CTP, and that reaction introduces the amino group on carbon number 1 of the pyrimidine ring. So now we have all 4 of our 4 of our triphosphates.